Okay, so this handout is my hand-drawn versions of the cells that you created using the bead simulation. And so what do you think the A's and the E's are in this diagram? What do you think they represent? Anybody know? No, we're looking at, we're kind of ignoring the sex chromosomes, so that's a good guess. So we're ignoring the sex chromosomes, so we're just looking at two, and remember the non-sex chromosomes are called autosomes. So we're looking at two pairs of autosomes. So what is a piece of DNA that is found in a specific location called on the chromosome? A piece of DNA that codes for a protein. A gene, right? So we're going to talk about that today, but the A's and the E's represent genes. And obviously, we have a lot more genes than just two. But notice how there are capital letters and there's lowercase letters. And so we're going to talk about variations in the genes that are called alleles. So we'll talk about that in a little bit when we talk about how genes are passed from one generation to the next. So that's what that represents. Now in this diagram right here, are there pairs of chromosomes? So this is the first question. Are there pairs of chromosomes in this cell? Yes, right? And I know that because they're of the same size and the same shape relatively, and I also have them listed with the same gene. So what are chromosomes that have the same gene called? Does anybody remember? Chromosomes with the same gene. They are called, oh, I lost that. They are called homologous chromosomes. And so these are chromosomes with the same size, same shape, and they have the same genes. Okay. So we have pairs of homologous chromosomes, except for the sex chromosome, because remember in the sex chromosome, we have the Y chromosome and then, the X chromosome and then the Y. So X and Y are actually non-homologous. So they would be said they're non-homologous because they have different genes on them. So that says R, non-homologous, the X and the Y. And we talked about one specific gene, which is called the SRY gene, which is found on the Y chromosome. And that is a genetic switch that turns the gonads into a testes. So the individual will look phenotypically and be phenotypically male. Okay, so let's see if I can get that image back up here. Let's see, where did my PDF go? Okay, so if we look at this one, it says, are these chromosomes unreplicated or replicated in this cell? They are replicated. And so you notice that when I replicate the chromosomes, that I copied the gene exactly. So that's why when I replicated that chromosome, I put two big A's. And when I replicated the other chromosome, I put two little A's. Okay? Because I'm replicating, faithfully copying the genetic material, and that is what is referred to as DNA replication. Okay? So is the cell above, this one right here, is this one haploid or diploid? Okay, so haploid means there's no pairs. So we know that there's pairs. So this is a diploid cell right there. Okay. So this is a diploid cell because it has pairs of chromosomes. Okay. Now we're going to talk about this in a little bit more detail, these two cells down here. Okay. But is this are these chromosomes replicated or unreplicated? They are replicated, right? And this would actually, would this be at the end of the first division or at the end of the second cell division in meiosis? The first division, excellent. Because in the second division, we get gametes, and that is what is shown here, right? So is this cell haploid or diploid? Haploid, excellent, right? There are no pairs of chromosomes. Are the chromosomes replicated or not? They are not replicated, okay? In this one, are there homologous chromosomes present in this one? No. There's a replicated chromosome, but there's not another replicated chromosome, which would be homologous, right? So we just have a replicated chromosome. There's not another one with the letter A on it. So they would be, the chromosomes are not homologous. The ones with the A and the E. Okay.
So even though they're paired?
They're not paired. They are stuck together when the DNA gets copied. Okay. So it's not really a pair. It's really a replicated chromosome that is going to get separated apart. A pair is completely separate. Right?
So if there's a pair that's completely separate and they're identical, then at that point they're homologous.
Yes, so in this one, this is a homologous, oh, I guess when I touch it, it doesn't show in this one. So when I touch the, if I circle the ones with the A, that's homologous. Those are homologous chromosomes because they're separate.
Even though one's capital and one's lower?
Yes, because it's the same gene, gene A. Yeah, that's a good question. Okay. So there's no homologous chromosome, but this is a replicated chromosome. So that's just some of the terminology that we're going to talk about and how this all relates to how you inherit traits from your parent. Okay. Okay, so we'll come back to this. So don't feel like if you're confused at that, we're going to come back to that. Okay. So in this particular image, and an image, a picture of the chromosomes is called a what? Karyotype. And you learned that you can do amniocentesis on a female that is pregnant, for example, and you can obtain fetal cells, and you can take those fetal cells and you can do a karyotype analysis to determine if, before the person is born, if they have any chromosomal abnormalities. You could also do a karyotype on you, like if you could take cells from your body and do a karyotype. Generally, they don't do that unless there's something they think is really dramatic. Because changes in the structure of the chromosome tend to cause big changes in the actual phenotype of the individual. Okay, so these chromosomes, if you look at them, are these replicated or unreplicated? If you look really carefully, I can see two arms. Okay, so this is actually a picture of replicated. When you did your karyotype analysis in lab, those chromosomes were unreplicated. Is this a male or a female? Female, okay. And this individual would be normal because they have two pairs of each. So we talked about when you get abnormal extra copy, and we're going to talk about why or how that occurs in a few moments. Okay. We also talked about the human life cycle. And so this is a diagram that shows that, and this is in your book, and I think this is in chapter like 5, can't remember. But this is actually in the cell reproduction part of the book. And it's drawn a little bit differently than we drew it. But you'll notice that the egg and the sperm, here, they do not have pairs of chromosomes. So they are haploid, right? And then they produce a diploid zygote. So we all start out as a single cell, a fertilized egg that is called a zygote. And then that cell undergoes mitosis to produce the multicellular organism that we are. And anytime we need to repair ourselves, Like if our liver gets damaged, the liver can regenerate itself if it stays, there's healthy enough piece of it, and that regeneration is due to mitosis. So the only place where meiosis occurs is in the ovary and the testes. Okay, so when we look at meiosis, let's go back to my meiosis. The things you need to know about meiosis is that there are 2 consecutive cell divisions. And in lab, we call these meiosis 1 and meiosis 2. Okay. Now, each of these have the same stages as mitosis, so prophase, anaphase, metaphase, telophase, but then there's a number. So if you see one with a number, like if you see prophase 2, you know they must be talking about meiosis. They're not talking about mitosis because there's no Roman numeral after the prophase, okay? The second thing that happens that's really interesting in mitosis is that during prophase 1, the homologous replicated pairs come together and they swap genetic information. The homologous replicated chromosomes the swapping of genetic information has a particular name, and it is called crossing over. Crossing over does not occur in mitosis. No crossing over in mitosis. So this is an example of genetic recombination, and that is actually the whole point of sex, is the recombining of the genetic information so that we get variable offspring. So that's important because in order for populations or species to change over time in response to infections or in response to different changes in the environment, there has to be variation. So this is our first example of the recombining of the genes. Remember that mitosis was asexual, no genetic recombination, whereas meiosis is sexual reproduction, so we have genetic recombination. So the crazy thing about this is it's like the shuffling of a deck. So you know how the chromosomes are sometimes red and blue? So if you had like a red deck of cards and you had a blue deck of cards, they would come together and then you would what is that called? By card. You would shuffle, okay. So you shuffle the deck, right? And then you pull them back apart. And then you'd see like, oh my gosh, there's totally different genes together than there were before, right? And so it's kind of like that. But there are some problems with that, right? So we could talk about the costs, right, versus the benefits. So the costs are that genetic mistakes can be made here. So you could get crossing over in the middle of a gene or in the middle of a piece of DNA, and then that gene would be mutated. And we're going to talk about mutations a lot. So that could be a mutated gene. And so when we think about crossing over, it's just amazing that it actually occurs. And so the benefit, we would say, must outweigh, so that's one cost. So the benefit must outweigh the costs, right? And that is that we produce variable offspring. Okay, so variable offspring are produced. Okay, so in your book, there is a diagram that shows this idea of crossing over. So if we look at the genes here, Notice that the blue chromosome has all of the big uppercase letters. And then the red chromosome has all of the lowercase letters. So if you were to think about what would happen if crossing over did not occur, right? A, B, and C would be inherited all together and as a group, and A, B, and C would be inherited together. So that's without genetic combination because we didn't do the shuffling, right? So notice what happens when we cross over. So this is why in your book and elsewhere, when you look at meiosis, only in meiosis will you see chromosomes that have different colors on them. So notice what happened when I crossed it over. Now I have A, big B, little c. And I have little a, little b, big C, right? So this means that this is going to produce more variation than that would have. Because I could have an individual that inherits big A, big B, and little c. Okay? So this just shows one incidence of crossing over, but it occurs up and down. Now, the X and the Y do not cross over. So that's kind of interesting because they are not homologous, right? They have different genes, and if they swap genes, then it would be really bad, right? Because we would not know what we were inheriting the next time we produce gametes, okay? So the other thing about meiosis that you should know, so this would be like the third thing. So the first is that there's 2 consecutive cell divisions. There's crossing over in prophase 1. And then in metaphase 1, we have replicated homologous pairs line up in the center of the cell. So this is important because it's the pairs. And so instead of them lining up individually, they would line up like this. So they would line up and then they would get pulled apart, right, by the cytoskeleton in the cell. Pull those to opposite ends. And so we end up with, initially, or after the end of meiosis 1, we end up with chromosomes that are still replicated. And to unreplicate them, to have the genetic material, then we would have to pull them apart again, which is in meiosis 2. Okay? So the idea of the pairs lining up, anytime you see them line up like this, you know that that is meiosis. Mitosis doesn't ever do that, okay? So I gave you a sheet that demonstrates that, but you probably have it in your lab notebook, actually. Okay, oops. But that demonstrated that. Okay, so this is in your book as well. So this shows metaphase 1. This is after metaphase 1, and then we have metaphase 2. And in metaphase 2, which would be in meiosis 2, the replicated chromosomes line up, and then we get the different gametes. Okay. Now, another idea about genetic recombination. Notice that these angiosperm, they don't designate which, have both blue, right? And that has to do with the way that the chromosomes lined up initially. But if they line up in a different way, so that there's a blue and a red on each side, then you can see that we produce different gametes. So the way the chromosomes line up, we'll say that the chromosomes will separate out differently into the gametes, and that is what we call independent assortment. So the fifth thing, maybe, or the fourth thing about meiosis is we have independent assortment. And what this means is that the chromosomes line up randomly. In metaphase 1. So what it means is that the chromosomes assort independently, so they have an equal likelihood of ending up with one chromosome or one type of chromosome with another versus the other type of chromosome. So an equal probability of having 2 red, right, or 2 blue, or a red and a blue, right? So that just means that the chromosomes line up randomly, and that is an example of genetic recombination as well. That one doesn't tend to have as much of a cost, though. Doesn't seem to have a cost at all, actually. It just increases variation. Okay, so I'll put increases variation in the gametes. Okay, so this is actually a diagram that shows the difference between mitosis, which is 1 cell, and you go from 1 diploid cell to 2 diploid cells, to meiosis. And notice if you start out with a single cell in meiosis, you could potentially end up with 4 gametes. So you start out with 1 diploid cell, and you could potentially end up with 4 haploid cells. Now, it is different depending upon whether you are male or female. Okay. So we're going to talk about the formation of eggs, which is called oogenesis. You say pronounce that O twice. And spermatogenesis. Okay. So let's look at sperm first. So we start out with a diploid cell. So this is my diploid cell. And this cell is in the testes. And it has been sitting in the testes, and it has been marked for a cell that is potentially immortal because it's going to get passed on to the next generation. So it is going to go through cell divisions. And as it goes through cell divisions, the cytoplasm is going to have, right? So that I'm going to get a smaller 2 cells, right? Because I have to have the cytoplasm. And then the cytoplasm gets halved again. So they end up kind of small. Right? Right? Like that. So the interesting thing about sperm is that in humans and a lot of animals, the sperm are actually even smaller than those 4 haploid cells. So these would be 4 haploid cells. And they actually shed what is left of their cytoplasm and become really small. So they have their nucleus, and then they have a little bit of cytoplasm, and then they have a mid piece, which has mitochondria in it, and then they have a tail, which is called the flagellum, right? So they actually get rid of almost all their cytoplasm. So they actually get energy from the fructose, which is produced by the male's seminal vesicles. So the male has, if we're talking about mammals, the testes are located out of the body cavity. Upon ejaculation, the sperm moves up. It picks up nutrients and energy like fructose. And then the sperm actually use the nutrients from the semen in order to power their ability to swim. Okay. So they're really, really tiny. So this is the sperm, right? Okay, so that's sperm production. That's spermatogenesis. So remember that we talked about how if you went to an alien planet and you were trying to figure out if you found a new species, if it was male or female, what would you look for? Do you remember? You take them back to your lab and you do some experiments and you would find out. How would you define which one's male and which one's female? Not genotype, because it could be a totally different mechanism of sex determination. Remember we talked about environmental, or we even talked about haplodiploid. Nobody remembers? Gamete size, right? Females produce large gametes. Males produce small gametes, okay? So we start out with our diploid cell, okay? And something really interesting happens during meiosis in the ovaries. And that is one of the nuclei gets all of the cytoplasm. Okay. The other nuclei gets hardly any cytoplasm. So I'll just put my nucleus and then it'll be a little bit of cytoplasm. Okay. So what they call this is called unequal cytokinesis. And it only occurs in the development of the egg. When we talked about the separation of the cytoplasm, in the males, it was equal cytokinesis, right? Because each nuclei got the same amount of cytoplasm. And in the end, they shed it anyway, but each nuclei got one, right? So here we have unequal cytokinesis, and this has a particular name, and this is called a polar body. Okay, so that's my polar body. And then this cell is going to undergo cell division, right, again, and it's going to produce, I ran out of room here, but it'll produce one big cell, right, and then it will produce the nucleus. So this is another polar body. That's a polar body as well. So I shouldn't have used a, okay. So this is a polar body and this is a polar body, okay. And what we end up with, we started out with one diploid cell, and we get one haploid cell, right? One diploid, one haploid. So we don't get as many eggs being produced as we do sperm, okay? And females in general have all the eggs that they're going to be able to produce when they're born, and they're kind of stuck in prophase 1, and then every month, a hormonal signals for one to continue through to finish metaphase, or to finish meiosis 1 and meiosis 2. And then it is ovulated from the ovary, and fertilization occurs in the fallopian tubes, right? So the main difference between males and females is that they are a limiting resource because it is much harder for the female. She produces a few eggs, but they have lots of nutrients. And in the case of like other animals like birds, right? But sometimes it's the male that helps, the male helps, but oftentimes it's the female that does most of the caretaking. And obviously in mammals, the female can't leave because she's the one that's lactating, right? So she kind of stuck with the offspring, right? In a mammal, not humans, but in other mammals, okay?
So the big cell is the haploid cell.
So this is the egg cell. So this is egg, the egg.
What happens to the polar body?
They are nothing. They just fall away. Now, the polar bodies can be used to sometimes detect whether or not the egg is normal or whether it has a genetic defect. So sometimes they sample the polar bodies when they are doing fertility treatments, and they look in the polar body to see if a particular mutation is present, and then they know whether or not the mutation is present in the egg. So we'll talk about that when we get to genetics, the genetics part. Okay. So if this is unequal cytokinesis, what is this? Equal, right? So it is equal cytokinesis. And that makes sense. Cytokinesis is just the separation of the cytoplasm. Okay.
So when is unequal cytokinesis taking place in the cycle? Is it when a follicle is made, when the ovaries become a follicle?
Yeah, so meiosis is really interesting. This is kind of complicated, but in meiosis, the follicle will divide, and then there will be one of the polar bodies. So meiosis 1 will occur during the formation of the follicle. The weird thing is that meiosis 2 is not completed after, and it is actually ovulated. The egg is actually ovulated, and meiosis 2 is not completed. And that second polar body is only formed after the sperm fertilizes the egg. So I don't understand why it does that, but it waits to finish meiosis until it actually is fertilized. And then meiosis occurs, or meiosis 2 finishes. That's a little weird. That's a weird thing. Okay. So when we look at errors in meiosis, oftentimes we can talk about non-disjunction. And this is where the chromosomes do not separate properly. Okay, so it results in what is called aneuploidy. Let's see if I can spell that right. Aneuploid. Okay, we'll put a Y on there. Okay, so if we look at this word and we break it down, this one's a really weird one. So ploid means copy. Eu means good. So euploid means that you have the right number of chromosomes. If you put an A in front of it, anything like asexual, then that means without. So this means without a good copy of the number of chromosomes, right? So without a good copy. A good number of copies. Okay. So for example, we can have what are referred to as trisomies. Right? So tri means 3. So you would have 3 copies. So what was the example of the trisomy? Actually, there was 2 of them, right? Trisomy 13 and trisomy 21, right? So those were 2 examples of trisomies that we saw in our karyotype analysis in lab. And also, we saw Klinefelters actually also. So we saw a sex aneuploid would be like X with an X chromosome. So we have, let's see, trisomy would be like 47, right? Plus 21, right? That would tell me it's the 21st chromosome, 47 plus 13. Okay. We can also have the sex chromosomes. So it could be 47 XXY, right? So this is what is referred to as a sex aneuploid, because we're talking about the sex chromosomes rather than just one of the other types of chromosomes we have. Now, the other thing that we can have happen during meiosis is we could have chromosomes breaking. So we could say that we have damage to chromosomes. Right? So in some instances, we have chromosomes that are missing pieces. And so a person who is a genetic counselor develops this search map for the chromosomes, and she knows exactly what she's looking for. And the reason why she knows exactly what she's looking for is that there's actually very few chromosomal mistakes that actually produce a viable embryo that you would actually ever be looking at, right? So if there's, so notice how we only looked at 21 and 13. If this was the first chromosome, which is huge, and you had a whole extra copy of all of those genes, you would never see that because that would be lethal. So these just happen to be non-lethal. It's not that they occur any more frequently than other non-disjunctions. It just happens that these are not lethal. Okay? So sometimes when you're looking at it, there are specific chromosomes that can be damaged a little bit, and that are sometimes observed, producing individuals that can survive, that might have some specific phenotypic characteristics to them. Okay. Okay, so in your book, they show this non-disjunction. Okay, so what this looks like is it could occur during meiosis 1. So in this particular instance, the chromosomes didn't separate, the pairs of replicated chromosomes. So notice that this, these gametes have 3. Yeah, so they actually have a diploid. So these chromosomes, this one and this one, are actually homologous, right? So they have an extra chromosome that should not be there. Okay. It could also occur in meiosis II. Right. Now, the reason why they think that Down syndrome is more common in females that are of a particular age, like over 37, is that for some reason non-disjunction becomes more common in eggs as a female gets older. So that's why if you're like 37 or older, then they oftentimes want to do genetic screening looking for the Down syndrome so that you know whether or not you want to keep the baby or whether or not you just be prepared for the baby being born with a genetic disease. Okay. Now the example of a missing chromosome, oh, here's my karyotype, right? So this would be a female with an extra 21st chromosome. Okay, so this is another condition that's sometimes seen. And so if a geneticist matches up the chromosomes and then she notices that there has been a deletion on chromosome number 5, this is an example of a disease that is called Cri-du-Chat. So here it is written down. So this is one of the most common syndromes caused by a chromosomal deletion. So it affects between 1 in 20,000 or 1 in 50,000, probably depending upon the population you're looking at. So unfortunately, this loss of that extra genetic material, causes a change even in their vocal cords. So that's why they call it Cri-du-Chat, which means cat. So their cry is more like the sound of a kitten rather than a baby. And then that was one of their characteristic phenotypic effects. Okay. So if we look at the symptoms of this, they tend to be small at birth and they have respiratory problems and their larynx does not develop correctly, so that is their signature cry. So there could also be effects in the body. So it's important to realize that phenotypes are not just what you can observe from the outside, but it also could be internal problems. So problems with heart defects, poor muscle tone. It could even be physiological, like they might have more than one protein, or they might lack a certain protein. And so that could affect their ability to survive. Okay, so we are moving on today, and this is something that we're going to be talking about and looking at next week as well. And we're going to have a lab over Mendelian genetics. So most of you have probably heard of Mendel. This is Gregor Mendel, and he was a monk that lived in the 1850s. And he is most notable for his study of pea plants. So in order to study how parents pass traits down to their offspring, he studied plants that he could collect seeds, peas, and then replant them over many, many generations. So he's rather meticulous with this. It's important to note that in the 1850s, they did not know what the genetic material was. They did not know about chromosomes. They could see cells, but they didn't know what was going inside of the cells. So really, he was just putting stuff into the black box in his experiments and then seeing what came out. But he actually didn't know what was actually going on in terms of what we now know in terms of chromosomes. Okay, so what I'm going to kind of skip forward and talk about as his research and then what we do know. Okay. Okay, so when you breed a particular plant, so we'll talk about pea plants, okay. You have traits and characters. So the traits here could be like height, and the characters would be tall versus short. And so he picked traits and characters that were very discrete. So notice that in us, height is very variable, and so we don't just have tall people and short people, right? We have, most of us are middle of the road in terms of height. But this is discrete, so there's only tall and short plants, right? And so it's pretty smart to pick something simple to work with, okay? So when we breed tall, and I'm going to put a little cross here, that means to breed short. Okay, so these are plants, pea plants. When we breed them, we are starting out with the parental generation, and we want to use true breeding plants. So this is true breeding parental generation. What that means is if I took this tall plant and I bred it with another tall plant, I would always get tall plants. It's true breeding. Right? It's never going to produce a short individual. If I took this short plant and I bred it with another short plant, it would always produce short plants. You would never get a tall plant. So that's what true breeding means, is you never get variation. You always have that characteristic, okay? So if I breed them, something interesting happened here is he didn't get a medium height plant, he got tall plants. Okay. So he's like, what is going on here, right? I always got tall plants. And this has a name. So it's not true breeding. So somebody in agriculture, you might be able to tell me this. If it's not true breeding, what is it? Close. Crossbreed, but it is also called a, see it on your plant seed packet. This is a hybrid, right? So this is what is called the first generation, the filial generation. So the first filial generation. And it has F1, right? And this is a hybrid, because it was produced from two true breeding parents, but it's a hybrid, right? So something really interesting happens if you breed hybrids. So if I take these tall plants and I mate them together by taking, he used a paintbrush and took the pollen from one plant and fertilized the flowers from another plant. So we crossbred them, right? If you take a tall plant times a tall plant, running out of room here, something interesting happens. You get tall offspring, and you also get short offspring. So what this means is that the hybrids are not true breeding, right? These are hybrids, and they are not true breeding because their offspring can be either tall or short. And then this one is called the second filial generation. So it is sometimes put with a big F and a 2 underneath. That's the second generation. So that was kind of the basis, and he used not only height, but he also used like seed color, pea color. He used whether the peas were round versus wrinkled. He used flower color. So there's many different characteristics that he bred. Okay, so this is an example: seed shape, round or wrinkled, seed color, flower color, whether the pods were wrinkled or inflated, right, yellow or green. Okay. So at the time when Mendel was doing his work, there was what it was referred to as the blending theory. Okay, so this is during Mendel's time. Okay, so this was what Mendel was testing. Okay. This was probably more like a hypothesis than a theory, but at the time it was thought that blending theory is how traits are inherited from parent to offspring. So in the blending theory, 1, offspring will always be intermediate between the two parents. And that makes sense because if you think about us, you know, our offspring are kind of a mix, right? So they're blended together, right? So the traits are blended together. Now, the other thing that was important is that once the traits are blended, you cannot separate them back out. And this doesn't quite make sense because if you think about, like, if you have 2 people with brown hair and then they have this red-haired baby, and oh my gosh, this is just like grandma, right? Grandma had the red hair, right? So that does not kind of follow what I intuitively know about how traits are passed from one to the other. And the other thing about the blending theory is because we're blending traits and we're always getting intermediates, is that variation was not seen as important. And we talked about how important we now know genetic recombination to be, right? And how we do produce very variable offspring. But back then, we didn't think variation was important. In fact, people were trying to find the most ideal form of each species, right? Not realizing that we have variation in a population that, like if we're talking about coloration and spotting on a moth wing, right? There's not an ideal version, right? The variation is significant. Okay. So if we go back to Mendel's experiments did not support the blending theory. So it did not support the theory of the time. And so that is why he is said to be kind of the father of genetics, right? Because he came up with a whole new idea of seeing how traits were passed from parent to offspring. Okay, so let's kind of fast forward and talk about what we know to be true about these traits that we now know as having this Mendelian pattern of inheritance, okay? So we'll talk about kind of the background. So remember that a gene is a piece of DNA that codes for a protein. We have variations of genes, and these are called alleles. These are not the term that Mendel used, but this is what we call them now. So alleles are variations in a gene, in a single gene. Right? And we now know that what he had experimented with was this idea that we have a dominant allele that can mask the recessive allele. So this masks. You can't see that the recessive allele there. So it masks, that's a K, the recessive allele. This is a capital letter. So if we had a gene and it was T, right, this would be a capital T. Then we have recessive alleles. and they do not mask anything, and they are a lowercase letter. Okay, so this is the gene. The dominant in this place is big T. The recessive is little t. Okay. So big T and little t. Okay, so let's go back to that experiment with tall versus short plants. Okay, so you might want to back up to your, sorry. Here we go. Okay. So what we are looking at here is the phenotype. So this is what the plant looks like. This is the outer expression of its genes, right? This is not the genes themselves. So we can define the phenotype as the outer expression of the gene. Remembering it could be inside a cell, but it is outside of the genome, okay? And then the genotype is the combination of alleles. Okay, so I'm going to change color here. Okay, so what is, do you think, what gene or what allele combination do you think the true breeding tall plant must have had? Dominant, just one copy? Two, right? So this would be big T, okay? What about the short? Little t. This is important because it's not an S. People want to put S's there, don't put S's there, okay? Generally, I'll give you the gene and then what it codes for, okay? So what do you think the hybrids had as their combination? Big T, little t. So that's a hybrid. Okay. Now the tall plants could have two dominant, or they could have a big dominant and a recessive. So these tall plants could be big T, or big T, little t. And you can't tell by looking at them. So that's why, for example, if you buy seeds and it says hybrids, you have to know that if the seeds get replanted in the next generation, you could get a lot of variation. So like if you wanted to have dark red hollyhocks, for example, and they're hybrids, and they say they're hybrid, I planted them, right, and I love the dark red, and then the next generation, they reseeded themselves, and I got yellow and pink. And it's like, right, and it's because I planted hybrid hollyhocks that I got variation in the next generation. Okay. So this one would be what? Little t. Okay. So one, it disproves the blending theory because this plant is not medium height. Okay. It was pretty smart to pick a trait that didn't have medium, just tall and short. So this is the first thing that it shows is that the offspring are not intermediate, right? And the other thing was that once these traits are together, right, in these tall plants, you shouldn't be able to get a short plant coming back out, but you do. So once blended, the short comes back out. And Darwin was actually a contemporary of Mendel, and Mendel's experiments would have fit really nicely to Darwin's idea that variation was important to natural selection. But they actually never got together. They were working independently. They died before they knew about each other's work. Okay. Okay, so let's look at another cross. And so let's look at albinism in humans. Now we cannot, experiment on people, and we also have a big generation time, so we could not produce all these different offspring, right, to figure out how albinism is inherited. So instead of using those types of experiments, we do what is called a pedigree analysis. We also do this with dogs and horses and other animals that have long generations, and you can see how the traits are passed down through generations. Okay. So in a pedigree analysis, a square is a male. A circle is a female. If this is colored in, okay, then this means that the person has the trait of interest. So I'll just put has trait of interest. And in this case, it's the lack of melanin in the skin. Okay. So I can draw a pedigree, and the first generation connected to the second generation, and then the third generation. Okay. So the way that I write the generations is with a Roman numeral. So this is my first generation, this is my second generation, and this is my third generation. And then I number the individuals. So I go 1, 2, 1, 2, 3, 4. Now notice that this individual right here married into the family. So they're not really part of the family, but they married in and they had a child. So we number that one, and then this is number 1. So for example, this individual right here is that designation. That would be generation number 1, individual number 2. Okay. Now at the moment, none of them have albinism, right, drawn in. So I'm going to draw in. Okay, and just by happenstance, it has to be that 2 females have albinism. Albinism is not sex-linked, so it doesn't occur more frequently in one sex than the other. So it's found on an autosome, not a sex chromosome. Okay, so pedigrees are really fun because you have to kind of view them as a puzzle, and you have to kind of look at all the pieces, okay? So in albinism, we have the gene A, right? The dominant allele is big A, and the recessive allele is little a. And I put that in there because it's always helpful for me to kind of like have a little legend so I can remember where I'm doing. Okay. So let's look at these individuals that have albinism. What must be their genotype? They haven't. What do you think? Do they have a big A? Oh, I forgot to tell you, did I? Sorry. The dominant allele is normal pigmentation. Sorry. Little a is albinism. There. Okay, so I always give you that much information. So what must be the individuals that are colored in? Little a, little a. Okay. Little a, little a. Can any of the other ones be little a, little a? No. Okay. Now let's look at their parents. So remember that this child had to have obtained a little a from each of his parents because that's the way it works, right? The egg and the sperm come together. So one A must have come from this parent and one little a must have come from that parent. So those individuals would have to be big A, little a. Now this is actually called a carrier, right? So we say that the parents are carriers because they carry the albino allele and then they pass it on to their offspring. But they didn't express it, right? They were normal. Okay. So what about these guys? We know enough about them to determine what they are? And I know that why? Yes. So this offspring right here tells me that those guys had to be carriers too. Okay. So that leaves us with this one. Okay. Now it could either be big A, or big A, little a. right? We don't know, but we could predict what the probability of it being big A, or big A, little a. So we can do what is called a Punnett square. Okay, so this is determining probabilities. So it's actually just a way to multiply percentages, actually, or decimals. Okay, so we determine probabilities in our Punnett square. So the male, it doesn't really matter, but we'll put the male on top and the female down on the bottom. And the male can either produce sperm with a big A or a little a. So that would be my sperm. So I could put them in my like little sperms. Okay. And then the female could produce an egg with a big A or a little a. I didn't line that up quite right. Okay. So if this sperm, which is a 50% chance, fertilizes this egg, which is also a 50% chance, then we get a 25% chance, or we get big A, right? Big A, little a. Big A, little a, little a, little a. Okay, so I'm just combining sperm and egg in the Punnett square. These are haploid cells. Okay, these are genotypes. Okay. So this genotype would give me, so this is kind of interesting. You could just ignore this, because remember what we're asking is, what is the probability that this individual has a big A, or a big A, little a, right? So we know they don't have that. So it's 1 out of 3. So it would be like a 33% chance that it is big A, right? And then like a 66% chance that it is big A, little a. So 33.3333, 66.66, right? 1/3 to 2/3. Okay. Now we could also use the Punnett square to ask other questions. So let's say they wanted another child. So individuals 2, 3, and 4 wanted to have another child, but they didn't really want to have an albino child. They wanted to know what the probability. So from that, what is the probability that you would have a child born with albinism? Looking at this. 25%, right? Because if I do this and I eliminate my crossed out this, so 1 out of 4 of their offspring is going to have albinism. And that is the same. So even if they had 10 children and all of them had albinism, really probability-wise, they have an equal chance of having another child. Because it is independent. Okay? So the last thing I'm going to mention here is that we have names for these genotypes, right? So if we have 2 copies of the dominant allele, what is that called? Does anybody know? Big A has another name. Homozygous dominant. So if you break down that word, zygous is the fertilized egg, and it means that they have the same copies of the gene. So homozygous kind of makes sense. And so that would be big A. Okay. Heterozygous. What do you think that one is? Big A, little a. Hetero means other. And then obviously little a is going to be homozygous recessive. And so that would be little a. Okay. So I think I'm going to stop there for today. And I think we're going to skip having the quiz. So we won't have a quiz on Monday. Everybody's like, yeah. Is that a good thing? Yeah.
On campus, it says that there's quizzes for and 5 too, Friday.
Is there 2 of them?
Yeah.
Then it probably is true. Yeah. I think they're simple enough, you'll be able to figure them out. I'll look and make sure.